Hello, biology students. Now we're going to do a practice of step 2 of protein synthesis called translation. This is very much a practice step because we are going to be using a tool that is not really the official tool. But it will look very similar. This box shape thing with letters and information in it is called a codon chart, but I'd ask that you label this as a practice codon chart. It'll work very similar, but the information inside isn't quite the same. Reminding ourselves from earlier that a codon is 3 letters in the mRNA, which is awesome because the way we look at this chart is that there are 3 major sides, and they're labeled here as the first side, the second side, and the third side. Where we can remind ourselves also of earlier, we were working on taking DNA strands and using the first step of protein synthesis called transcription, and we were using base pairing rules in RNA to match them up, noticing that there's U's here in RNA instead of T's. Well, the second step of protein synthesis is translation, which is taking the language of A's, T's, C's, G's, and U's, right, that were either in DNA or RNA, and really now moving towards a language that's meant to be a protein, because remember, this is all about making a protein synthesis. So our end result should be to make a protein. And proteins are made up not of nucleotides, but of amino acids. So each letter in this chart is representing an amino acid, which is the building block of proteins, a monomer of proteins. So if we look closely at this chart, these are actually not real amino acids. That's why it's a practice chart. But they are cool letters, or alphabetical letters, that will help us actually translate our language of nonsense and information that's a code, and make it into something that makes sense and is useful, because proteins are useful. So these alphabetical letters are meant to be useful. And again, codons are in sets of 3, so we're going to focus on the first codon, reminding me, is codon really in the mRNA or the DNA? Well, this hint was saying that it's in the mRNA. So I'm going to circle the first codon here, AUU, reminding us that codon is 3 letters, so the 3 letters in order are A, U, and U. And I'm going to look for AUU in my codon chart. So this codon chart is really nice since it's for practice. It says to me that the first side I'm looking at is on the left, the next one will be on the top, and the next one will be over here. So our first codon, I'm going to write off to the side to help us remember, is AUU. So the first letter is A. This side is A. So I recommend always that students put one of their fingers, usually their left finger, on this first side and look at the letters. And this is messy. This one says U, this says C, this says A, and this says G. So this first letter was A, so I put my finger on A. This means that I'm really focusing on this row with A. Okay, now the next letter, so we did A. The next letter is U, so I'm going to take a different finger, my right hand, and I'm going to look for on the top where it says second letter, because you used my second letter. I look for it, and noticing at the top it says U, C, A, and G. So the second letter is U. So that puts me here. Now if you've ever played the game Battleship, you know that Battleship, you say 2 letters or a number and a letter and you bring them together to meet. Well, that's how this will work. So because I've said this is the row I'm focusing on and now this is the column I'm focusing on, where do that row and column meet? Well, they meet here in this box. So now I'm really focused on this box only because that's where this and this meet. Now my last letter is U also, and I can see that the letters all repeat over and over again on this side. Well, if this is the box I'm mostly focusing on, because A and U met here, do you think I should look over here? No, right? Because this is the box I'm looking at. This is nowhere nearby. What about this one? Nope. This one? Yes, because this is the row I'm most focused on. Now here I'm going to look closely at these messy letters, and they're going in the same order over and over again. It was U, C, A, G. And the letter I'm really focused on, so we did A, we did U, and now we're doing the last one, U. And so U is right here. And so to pick out which is the correct one here, I move my finger across. I'm going to draw it this time, and this time alone, to get this. So all that work was to get this, which is representing one building block in a protein, an amino acid, and I'm going to write the letter E up here under that codon. So I've now done translation. I translated all of this into a single thing, an amino acid represented by this letter. The next codon I'm going to do is GAA. I'm going to follow the same process, which will go faster and faster. G is here for the first one. Let's write it out, GAA. The next letter is A. Where do they come together? I'm focusing on this row, and my second letter is A, this column. Where do they meet? Oh, they meet right here. Somewhere in this row and this column meet here. The last letter in GAA is A. So I look on the far right side, UCAG, GAA. Here's A, and I bring it across into the box I'm most interested in, and that selects X. So that was a lot faster now that we're getting how to do it. Let's do another one together. U. So for U, which is kind of fun to say, I am in this, I'm in this. They meet over here. And my last one is U, and that takes me to the one I pointed out earlier, which is A. Let's keep going through this whole first one. You might be able to guess what it is. Don't yell it out if you're in class. The next codon is ACC. A, C. They meet, and I need to pick C for the last one. I bring it across. That takes me here. The next codon is GCG. GC meet in this box somewhere. And the last one is G, right? Double check. Because if I pick the wrong letter, I'm going to get the wrong translation, which will make a typo or a mutation. So the last one is G. That gets me this guy. I wonder what it is. The next one is CCA. CCA. This is hard to read. These are L's. This is the letter K. All right, so L, what could it be? What could it be? A-U-C, A-U, A. Did I do it right? Because if I did it wrong, that would be when maybe something would be messed up here. A-U-C. Huh? Nope. Sounds pretty good. Well, what's going to be the last thing? Because that looks like a pretty logical word for this thing. So funny. A-G-A. A-G-A. That takes me here. This is an exclamation mark. Yay, we did it. Now, you should try to do these other ones. Some of them are jokes, and some of them are words or people that we've learned about. Notice that there is some of these that say space. If for some reason something's not working and it's not making a sentence or a word that makes sense, you probably did some typo when you're in the middle of your transcription or your translation, and you should maybe relook at it carefully before getting too nervous. We will look at a real codon chart in a future class, but great job practicing this, guys. See you next time.